methylated promoters, but in general promoters seem to be highly unmethylated, so that they can be transcribed, of course. While when you look at gene bodies, you have two peaks here. So you have unmethylated gene bodies, but you also have a lot of methylated gene bodies. You can see you have a bimodal distribution right here, which is statistically a little bit difficult because, well, statistically thinking, we have very well established methods to measure gene expression and to measure the differences between conditions, where you always have a kind of normal distribution, which is always nice when you're doing something with statistics. In methylation, it's a little bit more difficult because you always have these bimodal distributions. So how is a typical microarray analysis workflow? First of all, in whatever you do, you have to do a quality assessment. This is really important, and some people do not, in my opinion, do not pay enough attention to this part of the analysis. Because if you have some low quality probes, or even worse, low quality samples in your data, because there was something wrong in the experiment, because whatever could happen, and you do not remove these samples from your analysis, you measure whatever it is, but not a real condition you want to look at. So it's really important to remove all data which might be suspicious, because it influences a lot what you see later on in your analysis. So what you do then is, you always have to normalize your data. This way, we are trying to reduce effects of experimental variation, which you can never exclude completely. So you just try to make your samples comparable as much as you can. You try to adjust for background, which is, whenever you measure, you always have some technical background signal which comes from the scanner, from reading the intensities. So you always have a background level of noise, and you want to reduce this, of course. And what is done in general in methylation data commonly is just an average normalization, because you would expect that the average methylation across all your samples in average is similar. So the next step you can do is just to visualize your methylation data, to explore it a little bit. What you could do is a kind of scatter plots to compare samples with each other, to do just some clustering or heat maps, as you can see here, for your different conditions, where you might already see differences in general methylation patterns. Or here you can see a classification where you have different conditions you were looking at. You could do a kind of circle plots, where here you have darker values for one sample here, which is more methylated, and the lighter color is less methylation, and here you can see already a little bit what is going on. So there are many different ways of visualizing your methylation data. But what, of course, you then want to do always is to identify differential methylation. So you want to know which are the positions or regions which have different methylation patterns between different conditions, such as, for example, disease versus healthy, or you're investigating different tissues or different cell types, and you just want to compare different individuals, whatever. But it's important here you employ whatever statistical test which might be appropriate for doing this. You have to bear in mind that when you do this, in microarray analysis, you look at a lot of genes parallel. You're not doing just one test for one gene, but probably you're doing a test for about 22,000 genes. So classically in statistics, what you would have is that you say that if from your statistical test you get a p-value of 0.05, you would regard it as significant, as you probably all know. But when you test around 20,000 genes, and imagine that the null hypothesis is true for all of them, then you would expect to have altogether 1,000 false positives. So you would say that there are 1,000 genes with differential methylation, also there is not any gene with differences. So it is really important to do an adjustment for this multiple hypothesis testing, as we call it. There are several algorithms working with this. What we generally apply is Benjamini-Hochberg correction. You can also look it up in literature how this is done. Hochberg, yeah.
Okay.
It's quite a famous method which is also used a lot in gene expression studies, etc. It is very important to take this into account because otherwise you report a lot of changes which are not really significant statistically. And in the end, what you do, of course, is you just rank your results in terms of statistical significance and probably also in terms of effect size. So if you have a region or a gene where the beta value changes from 0.01 to 0.015, maybe it's not that interesting. So maybe you also want to compare what is the range of change which is occurring. But of course there can also be some very minor changes which are statistically highly significant and could also have an influence. So you also have to think about how you want to do this. And finally, of course, you want to interpret the differences in your DNA methylation. What we're doing a lot in gene expression analysis, for example, is when we finally have identified genes which are differentially expressed, we just do some kind of gene set enrichment analysis, it is called. And we want to know that if, for example, all the genes which seem to be important for our condition are genes which are all participating in a kind of the same pathway or something, so that this way you can know what are the important functions or pathways implicated in your conditions. This is very well established in bioinformatic analysis of gene expression. But when you're looking at methylation data, you have to be very careful with this, because you cannot apply the same algorithms you use in gene expression results than on methylation, because what happens is that the probes on the microarray per gene for methylation, they are not evenly distributed across different classes of genes and gene promoters. This is also because of the distribution of the Cs, etc. So if you would just apply the same methods without taking this into account, you also using just the random data, you get significant results because of how the microarray for methylation is constructed. So there is a paper, if you're interested in this, where they are explaining how to solve this problem. So never apply directly methods established for gene expression analysis, for gene set analysis to methylation data. And of course, what you also might want to do is just some kind of integrative analysis in the context of maybe other data sets you already have analyzed. Very important. Whatever bioinformatic analysis you're doing on biological data, you will always have batch effects. Something you do not want to have, but you cannot avoid. What is a batch effect? This is a confounding of between sources of technical bias, which is the batch effect indeed, and the phenotype of interest. So for example, what could happen? You could have hybridized the samples of your different conditions on different days maybe. So there could be people who do the normal samples on one day and the tumor samples on another day. But then in the end, how will you know if there was just, for example, a change in room temperature of 1 degree or something, which influenced your results already? And the same with utilizing different preparation kits or with different microarray benches or whatever. So you always need to try to avoid as much as possible to have such confounding variables. So you should do all your experiments at the same time point with the same material, following the same protocols, etc., etc. But of course, there are also some possible non-technical confounders which could cause batch effects as well. So if you have one data set where you only have males and another data set where you only have females, and one of them is cancerous and the other one is normal, how will you know if it was just sex difference or if it was because of the disease? So you have to take care that the distributions of the different conditions are equal in your samples in respect of sex, age, ethnicity, whatever. Interestingly, what has also been shown is that even if you take, for example, samples from humans in the morning, the results are different from taking samples later in the afternoon. So never confound these things then with your condition you want to investigate. So there are also some tools which aim to remove such batch effects, but of course if you have a lot of confounders like this, it is impossible and you should try to avoid it as much as possible before not to, and because tools cannot do any magic around. Of course, there are also some limitations in microarray analysis. First of all, what happens in microarrays is also cross-hybridization. So there happens a non-specific binding of DNA fragments to multiple probes, which then causes false positives. So you have your probe, which is just a fixed length of your sequence, and maybe if there is just one or two bases which are different, you could have this sequence binding, although it is not the same sequence actually. So this is happening in microarrays, and you have to take this into account. What is also happening is that genetic variants are affecting probe binding, above all SNPs. So SNPs, I already mentioned them briefly today in the morning. They are single nucleotide polymorphisms, as you can see here, where one base gets changed, which you could also call mutation. But as it is present then, because it was a successful mutation, not causing damage or even being advantageous, it is established then in a significant proportion of the population of a species. So therefore, in some populations, you might have different of these point mutations, which then influence your measurements in microarrays if these sequences are the ones which are in the oligonucleotides which are on your microarray. So what I generally do is that I remove such probes from my analysis before doing something, because it would cause problems as well.